Welcome to Savannah Flowers Laboratory. Here is brief overview of our laboratory. First, we visit the rooms. Then, we go into the details. We have 2 growth rooms, initiation room, storage room, media preparation room, thermotherapy room, virus testing room, and PCR testing room. When entering the lab, we first change our shoes, leave personal shoes outside the facility, then wear lab shoes, thoroughly wash hands, wipe hands using clean towel, sterilize using sodium hypochlorite, wear disposable or reusable head cap, put on clean lab coat from the hanger. Sterilize hands on the table using ethanol 70%. Wear surgical gloves, mask. Sterilize again, then you are ready to proceed in the other rooms. We have a screen that controls the lab rooms. For preparation of solutions, we always use a checklist to eradicate errors. We accurately weigh components using analytical weigh balance. Technician, adding sucrose in conical flask that is placed on a magnetic hot plate stirrer, adding PGR using micropipette. Then we sterilize the containers for media using an autoclave. We place the flask on a magnetic hot plate stirrer. Then we accurately measure different stock solutions using volumetric flask, and then we add into the flask for mixing. After mixing, we add distilled water to the desired working media volume. Lastly, we rinse the pH meter using distilled water, then check the pH, put in the heating rod in the flask to heat the media. Then add solidifying agent and leave the media to heat as it's stirring. Take the conical flask containing media into autoclave to sterilize. Take the media to the laminar flow. Disinfect surfaces using 70% ethanol, and then pour media into containers aseptically and leave to cool. CHOD initiation, start by sterilizing tools in the autoclave. Collect explants in sterile containers and label the containers. Wash using distilled water 3 times. Add few drops of tween 20 and place them on electric shaker for 17 minutes. Rinse 3 times using distilled water and surface sterilize the containers and place them in the laminar flow hood. Add ethanol 70% for 30 seconds to 3 minutes depending on explant type. Rinse 3 times using double distilled water. Add 10 to 20% bleach solution and leave to stand for 10 to 15 minutes. And then rinse the EX plants using double distilled water 3 times, after which the explants are ready for initiation. Set up the laminar flow. Place the explants on the stage of microscope and dissect to obtain meristem, difficult to observe with naked eye, and then place the meristem in initiation media, label, and take to growth room. Multiplication, sterilization, same as initiation. Pick the plants from the growth room, surface sterilize before placing them in laminar flow. Place plants on sterile paper and use sterile tools to multiply plants and place them in multiplication media. Seal the container. Label and document will be used to enter information in TC database. Take sample from the cultured leaves, chalk stem, in a sterile sample bag for pathogen testing so as to know plant status. Growth room. Shelves are labeled for easy identification. Different plant varieties are placed in designated shelves. Data loggers are placed in growth room for secondary temperature check. Routinely, technicians check plants for contaminations and growth progress. Primary hardening is done in the shelf with double lights before rooted plants are taken to propagation for hardening. Acclimatization, secondary hardening is done here. Plants are sticked in plugs and placed under a moist fleece cloth for them to grow. Plants stay here for 4 weeks. Documentation. We have 3 main documentations. 1. Database structure. Has all the information regarding the plants from initiation to acclimatization. The information can be exported to Excel for data analysis purposes. 2. Production projection helps in plant output projection factor. 3. In vitro follow-up. This document helps us in detail when plants in growth room are ready for processing. At the close of business, the UV lights are put on in various rooms using a remote controller to sterilize the environment and make the space ready for the next day.